Hello everyone, this is Tariqul Haque Tuhin. I'm a postdoctoral fellow at Department of Pharmacokinetics and Drug Metabolism in Amgen in South San Francisco side. Welcome to my talk at the Drug Discovery and Development Virtual Event Series 2025. Today I will talk about protein engineering strategies to overcome charge-driven clearance liabilities of monoclonal antibodies. I will start with a brief introduction about monoclonal antibody clearance. The disposition of immunoglobulins is influenced by target-independent and target-dependent mechanisms. Target-dependent mechanism is dependent on the target biology and manifests due to the affinity and selectivity of monoclonal antibodies to the target. On the other hand, target-independent mechanism is important when the target route is saturated or not a driving factor for overall PK. This route is governed mainly by non-specific endocytosis and FcRn-mediated recycling. I will talk more about these two cellular processes because they determine the target-independent clearance. So IgG internalization without receptor is mainly driven through non-specific endocytosis, which is mediated by both adsorptive and fluid phase endocytosis. It is also mediated by neonatal Fc receptor, or otherwise we call it FcRn receptor. FcRn receptor diverts internalized IgG and albumin away from lysosomal catabolism via pH-dependent interaction. That is why inclusion of human Fc domain is a very well-validated half-life extension strategy. There are two examples in this figure with a low target-independent clearance map and high target-independent clearance map. For a low target-independent clearance map, initially there will be low non-specific endocytosis, and as a result, there will be, when the map is internalized, there will be low non-specific interactions at acidic pH and good human FcRn capture. It will result into low lysosomal catabolism and high recycling efficiency. For high target-independent clearance map, initially there will be higher amount of non-specific endocytosis. There will be high non-specific interactions when it is internalized at the acidic pH, and as a result, there will be poor FcRn capture and lower recycling efficiency and high lysosomal catabolism. I'll talk more about the non-specific endocytosis as this is a predictive of target-independent clearance. We have developed a CHO cell-based assay that can successfully identify elevated target-independent clearance. We have tested 127 proteins that spans with a clearance range from 0.1 to 10 ml per hour per kilogram. And in our CHO cell-based assay, it could flag 75% of the proteins with target-independent clearance greater than 0.5 ml per hour per kilogram. So when we tested this assay along with other 20 biophysical assessment, this was the most capable in vitro assay for predicting target-independent clearance. With this assay, we have the ability to screen hundreds of thousands of proteins per week within PKDM. Now I will talk about cellular human FcRn recycling assay. We have developed this assay within PKDM, and this assay utilizes two cell lines. One is the parental MDCK2 cell line, and the other one is human FcRn expressing MDCK2 cell line. With this assay, we collect uptake samples, recycled samples, and residual samples. We have established a scoring metric. We call it frame scoring, which is FcRn recycling efficiency metric. This metric is dependent on two major things. One is the FcRn recycling efficiency, and the other one is non-specific uptake coefficient. We have tested 8 monoclonal antibodies in this assay, and as we see here, the higher the frame score, the lower is the clearance, and when the clearance increases, the frame score gets lower. So when the frame score is below this red line, which is 0.25, we see these molecules have high risk for human target-independent clearance above 5 ml per day per kilogram. So the takeaway from this section is accounting for both non-specific assessment and FcRn engagement within the same assay can increase PK predictive capacity. Now I'll talk about two molecules that we use as our tool monoclonal antibody within our department. One is the previous clinical candidate of Amgen anti-IL-4 alpha mAb. It has a very high clearance in human, 11 ml per kilogram per day, and it was targeted against IL-4 alpha receptor. The other one was ASA. It's a anti-streptavidin antibody. It's a tool antibody within our department. And when we tested these two molecules in mice PK, we saw that anti-IL-4 alpha mAb exhibited very high amount of clearance compared to ASA mAb. And when we investigated these two molecules in our two cell-based assay, we found that anti-IL-4 alpha mAb exhibited very high amount of non-specificity compared to the tool mAb ASA. When we performed homology modeling, we found out that there are 4 positive charge patches because of arginine in the anti-IL-4 alpha mAb in the variable domain. The overall variable domain net charge at pH 7 for anti-IL-4 alpha mAb is +7, whereas the net charge is much lower for anti-streptavidin molecule, which is +2 only. So as we see here, the charge impacts cellular processes and it also impacts the target independent clearance. So here is a mechanism we are showing with a high target independent clearance map. So with the high target independent clearance, initially there will be rapid non-specific adsorptive endocytosis, and when the molecules will be internalized, there will be a pH-dependent charge jump because of endosomal acidification. As a result, there will be limited FcRn engagement and low recycling efficiency. So as a net result, there will be overall uh, lower FcRn recycling efficiency and higher target independent clearance. And as we see here, charge is not only important outside of the cell, it is also important inside of the cell as well. We have performed homology modeling of anti-IL-4 alpha mAb and with some mutated molecule that we have reported earlier. And here are examples with anti-IL-4 alpha wild type mAb and the EES mAb, which is a mutated mAb. And for EES, we have mutated the 3 arginines with glutamic acid and 1 arginine with serine. If we look at the first figure on the left, we see here over a range of pH, the variable domain net charge of wild type molecule is higher compared to the EES molecule. And as we look here in our two cell-based assay, we see when we mutated the exposed positive charge patches, the overall non-specificity of the molecule decreased significantly. We tested the wild type and EES molecule in TG276 mice PK, and we have found that the EES mutant mAb exhibited very high amount of serum concentration profile compared to anti-IL-4 alpha mAb, which exhibited very high clearance. So it is important to assess charge character throughout endosomal or lysosomal pH range to rationalize intracellular trafficking. Now I want to talk about the half-life extension strategy of monoclonal antibody. One of the very common strategy is YTE mutation. It's a triple point mutation in the CH2 domain of the monoclonal antibody Fc region. YTE mutation is optimized and it has increased FcRn affinity, and it's a clinically proven model for half-life extension and for decreasing target independent clearance. Here is an example with MEDI527 YTE molecule and MEDI524 molecule. As we see here, the YTE molecule has lower clearance compared to the MEDI524 wild type molecule. So now we have reached a couple of key questions. How much non-specificity can these YTE maps overcome compared to the parental wild type map? And which one is more impactful? Is it decreasing the non-specificity or introducing YTE at the Fc domain? And can we quantify the potential benefit of YTE incorporation via in vitro cellular approaches? So we have expanded our previous library we have engineered the variable domain as well as also the FC mutation we have introduced, and we have now some single point mutants, double point mutants, as we have mutated the arginines in the variable heavy chain domain, also in the CDR1 and CDR3, and we have one more mutation of arginine in the variable light chain domain. We have total 10 pairs of AMG317 wild type and YT map mutants. So the overall goal of the study is to survey both map, FV, and FC domain engineering strategies to overcome variable degrees of non-specificity and decrease target-independent clearance. So first we started with the Joe cell-based non-specific assessment. As we see here, even with a single point mutation like in 20S, which is introduced in the variable chain, heavy chain variable domain, the non-specificity of the molecule decreased significantly. But when the single point mutation was introduced in the CDR1 or CDR3, the amount of non-specificity decreased was more significant. And with the double point mutation, like as 33S and 110L, we see that the non-specificity decreased even further. And all the triple and quadruple point mutations, they also decreased, demonstrated significant amount of decreased non-specificity. We have tested the non-specificity at both pH 7.4 and 5.8, and we have found that the YTE counterparts also exhibited lower and lower non-specificity with more mutations. So the takeaway from this section is if we reduce the surface positive charge, we can reduce the non-specificity of the molecule. Then we tested 6 pairs of molecules in our FcRn recycling assay. And as we see here, the wild type and wild type YTE molecule exhibited very low frame scores. And when we introduced single and double point mutation, the frame score increased significantly. The highest frame score was achieved with SSS YTE. Overall, if we look, all the YTE mutants exhibited higher frame score compared to their wild type counterpart. And with more and more mutation, we have seen that the FcRn recycling efficiency metric of the molecule increased significantly. So the takeaway from this section is YTs exhibited improved cellular recycling efficiency when compared to their parental wild-type maps. Next, we performed PK studies with these 6 pairs of molecules. Initially, we performed a small-scale TG276 mice PK study, and we have encountered some anti-drug antibody presence issue in that PK study. So we opted for an immunodeficient mice model. So we have used the SCID TG32 human FcRn mice for our PK study. And as we see here, the wild type anti-IL-4R alpha map exhibited very high amount of clearance as exhibited, and the wild type YT map improved the clearance, so it exhibited lower clearance compared to the wild type map. And when we introduced the point mutations to mutate the exposed positive charge in the due to arginine, we see that the overall PK profile of the molecules increased and got better, and the highest serum concentration PK profile was achieved through the SSS and SSS YT mutation pair. And so far, as we see here, that the 33S, 110L, SSLS, and SSS, these 3 map mutants resulted in very improved PK. So the improved YTE PK performance is more prominent in maps with lower non-specificity and altered FcRn interactions. If we compare the frame score to the clearance value, we see here the higher the frame score, the lower is the clearance, and if the clearance is high, the frame score goes down. So if we look here, the wild type molecule has very high amount of target independent clearance, and it has the lowest amount of frame score, and the AAAS YT mutant map that has the highest frame score and has the lowest clearance among all these molecules. And if we look on the graph on the right side, if we can see that with this cell-based FcRn recycling assay, we can capture the reduction in YT mouse clearance percentage. And as we see here, the SSLS molecule demonstrated very minimal amount of uh, serum PK profile improvement with the YT mutation. So it was the lowest among them, but the other mutants with the YT molecule that improved a very high amount of serum PK profile improvement compared to their wild type counterparts. So if we account for both non-specific assessment and FcRn engagement in the frame scoring, it can increase our PK predictive capacity, and reducing non-specific endocytosis is more impactful on target-independent clearance and frame scores compared to the YT mutation alone. So the summary from this talk is the target-independent clearance processes can be a significant driver for PK variability across MABs and other biologics modalities. Cell-based assays can be utilized as powerful PK predictive tools, as we exemplified from our studies. Reducing non-specificity is more impactful on target-independent clearance than the YT mutation alone, and incorporating both the FV and FC mutation resulted in better frame score and reduced target-independent clearance as observed with the YT mutants compared to their wild-type counterparts. So here are some applications of the cell-based assays. Cell-based assays can guide discovery or early optimization campaigns. We can identify high-risk biologics in early development easily with the cell-based assays. We gain important mechanistic insights, and we can predict the success of MAB HLA engineering campaign with the cell-based assays. And with these assays, we can minimize the utilization of resources for in vivo experiments. I want to thank my colleagues in PKDM, Discovery Protein Science, Research Biomics, Process Development, ASF Fluorocore, and as well as Postdoc Steering Committee. Especially, I want to thank Cape, Mark, and Caroline for their support and guidance in these projects. Thank you, everyone, for attending this talk. If you have any questions, please feel free to reach out to me. I'd be happy to chat more. Thank you.